So hello everyone and welcome to today's webinar. I'm just gonna put my slides back up there again. My name is Dawn Van Dam and I am the Executive Director of 1 Million Solutions in Health. And I wanna welcome everybody to today's presentation. It should be phenomenal and very interesting and informative. And I encourage everyone to ask tons of questions. So in one moment, I will get Zoom all set up. Oops. There we go. Great, awesome. So today we're going to be sharing an NIH licensing opportunity called a novel, highly effective HIV vaccine. It's made more effective due to a multi-component approach to preventing and treating HIV infection. We'll have a bit of welcome and introductions. We'll have our presentation, signature square presentation by Dr. Genoveffa Franchini, and we'll ask you to do a quick evaluation and have some closing remarks as well. Our vision at 1 Million Solutions in Health is to accelerate the discovery, development, and delivery of innovative cures, treatments, and preventative measures for patients around the world. We do that through our Hidden Gems program, where we help you find science or get your science out there to the people who can help commercialize it. We do that by working with the industry, NIH, academia, and entrepreneurs. We also have had a technology evaluation consortium where we work with the pharmaceutical and biotech industry and technology vendors or scientists to, again, make those connections and bring the science forward. And this is just a sampling of some of the people that we have worked with in the pharmaceutical and biotech industry over the last number of years. And we're very proud to have those relationships and can help bring you all this great science. So fundamentally, we help find new scientific solutions and gather input to optimize the technology and then share the findings through platforms like today's webinar. So a little bit more about today's session. This webinar will incorporate 3 technologies that work independently, but when combined, result in a higher level of HIV vaccine efficacy. The first technology is a V1 region deleted GP120 modified protein that shows an increased immune response to the V2 region and superior vaccine efficacy. The second technology demonstrates that increasing RAS activation results in increased vaccine efficacy. The thought here is to combine a RAS activation agent with the first technology's V1 region deleted GP120 modified protein to further enhance the vaccine's effectiveness. The third technology demonstrates how a recombinant DNA and an ALVAC vector can be used to manufacture a more potent vaccine candidate. The thought here is to use this method to manufacture the V1 region deleted GP120 modified protein to further enhance the effectiveness of the resulting V1 deleted GP120 vaccine. So a little bit about our presenter for today. I'm just going to skip to that here. So Dr. Genoveffa Franchini, she's a senior investigator at the vaccine branch, the head animal models and retroviral vaccine section. And she's in the National Cancer Institute's vaccine branch at the NIH. She is a hematologist and renowned retrovirologist who has pioneered research on oncogenes and human retroviruses. She has made numerous achievements in virology and translational approaches to prevent human diseases caused by retroviruses. Her work has furthered the understanding of HIV and HTLV1 pathogenesis, leading to the identification and characterization of new viral genes for HIV and HTLV1 and their functions. Her accomplishments in HIV vaccine development include the preclinical work that led to the first vaccine trial in 16,000 volunteers in Thailand that has demonstrated significant, though limited, protection against HIV acquisition. Her basic work in immunological mechanisms of protection furthered the understanding of the efficacy of the currently available smallpox vaccine in primates. She has also pioneered strategies to down-modulate regulators of immune response in HIV-1 infected individuals using the Mackey model of SIV infection. So with no further ado, I'm going to stop sharing my screen and let Dr. Genoveffa Franchini bring up her presentation and take you through it today. Again, at the bottom of your screen, while she's getting her presentation up there, is a chat box. And if you could put any questions that you have during the presentation into the chat box, then I will ask Dr. Franchini to answer them at the end of her presentation. So keep the questions flowing and we'll take you from there. So Dr. Franchini, if you could take yourself off mute and share your presentation, that would be awesome. And over to you. Thank you.
Okay. Hello. Hi. Good morning to everybody. So following this excellent presentation on the overall goal of my presentation, I would like first to stress that and make you aware of the model that we have used to obtain the results that I will present today. So the animal model is a small non-human primate model with the rhesus macaque. And we are using a virus that is a distant cousin of HIV and is able to induce the disease AIDS in 90% of the animals. It's a very rigorous model, and therefore, having a vaccine that can protect against infection probably is highly significant for humans. And in fact, this animal model has so far predicted the results of actually all of the 6 human trials done, 7 actually human trials done, and also has recapitulated them. So I'm pretty confident that some of the results that I will show you might really be relevant to humans. So let me just go to the need for an HIV vaccine is exemplified by the number of people that have been infected, which is around 40 million people. And is not resolved the problem at all, even if it is perhaps less talked about than it was in the last 20 years. And still there are approximately 5,000 people that become infected every day worldwide. And of those, approximately half are young adolescent female in sub-Saharan Africa. And for these people, they don't really have an opportunity to have access to the therapy. And the therapy, the current standard of care, as you know, is constituted by several small molecules that target the receptor and a variety of other targets, but also alone or in combination with the antiviral therapy that really targets viral enzymes. And it is highly effective, but as we said, many people don't have access to this therapy. So the target of the vaccine is obviously to try to eliminate the infection and then eliminate the need for lifelong treatment. So the solution, as we said, it could be a vaccine because the vaccine can eliminate infection, can prevent infection, and then it might be given a few times. Maybe you need some boost, like for many vaccines that we are using now for other diseases, but it's not a long time, it's not a lifelong treatment. And it might even change, should this also work as a therapeutic, the standard of care for maintaining the virus under control in people that are already infected. So currently there are, I need to move, okay, because I can't really see. Okay, so currently there are several vaccines that are in the pipeline, and most of them, and not only vaccine, some of them are also synthetic peptides, but some are also not exactly vaccine, but monoclonal antibody already made and given that can prevent infection. And they're being tested now in human trial. But most of them really target the GP120, which is the surface protein of HIV. And but none of them are actually using what we are using, which is to create an immunogen, which is now devoid of some unneeded and perhaps possibly deleterious epitope, which can be recognized by the immune system and help the virus rather than to help the host, as I will show you afterward. So the three technology that I'll be talking today about, as Dawn has already indicated to you, can be combined together to induce antibodies to V2. The V2 region is expressed on the surface of the virus. V2 stands for variable region 2. Is presented on the surface, so is immediately seen by the immune system. So antibodies against this region have been found as a correlate of risk in the only human trial that has actually given some results of protection. And these antibodies against V2, together with the epigenetically modified monocytes and T cells, we believe are really important in giving protection against HIV. So we have moved this technology to a phase 1 clinical trial. The NCI is supporting a phase 1 trial with the component of these vaccines. And particularly, we are combining the technology 1 and the technology 3. So how did we get to this? As I said, we used hundreds of animals to try to understand why the vaccine worked, but also why the vaccine didn't work in some of them. So what we found, and this is the basis of the technology number 1, is that as you can see here on the top, on the left, you have a picture, a schematic picture of HIV, of the virus. What you can see here highlighted with the red is a trimer, which is the envelope of the virus, which is forming this, basically 3 molecules of this envelope are associated with each other. This is very important for the virus entry in the cells. On the bottom here, you can see a cartoon of the structure of the virus, and it shows you where the V2 is, and is the red and the green are 2 different parts of the V2, but it doesn't really matter. Is the old V2, as you can see, is in here, and is underneath a kind of greenish area, which is called V1. So you can already see that if this is what the immune system is seeing, it might not be very easy to see the V2 if you have the V1 on top of it. But in addition to that, if you also add an antibody on top of this, you might actually mask the V2. And it might be impossible for the antibodies that are cytocidal, that bind to the V2, to protect. So what is the basis of this possible protection? The antibodies are cytocidal, so they see the infected cells because they can bind to the surface of the cells and they can kill them. But also these antibodies are able to block the ability of this V2 region of the virus to bind to the alpha-4 beta-7 integrin. The alpha-4 beta-7 integrin is very important for the cells to migrate to the gut and to other organs. So this is the way in which the virus probably spread. So if you have antibody that block also this interaction, not only kill the cells that are infected, but also block the interaction of virions, because now they can't attach to the cells. You can imagine that this could actually contribute to protect from infection. So how did we get, as I said, to this? We get it just looking on the right here. You have a little Kaplan-Meier curve that shows you that animals that were not protected from HIV, or in this case, HIV infection, were the animals that had the highest response to the V1 region. So this led us to hypothesize that perhaps the removal of V1 by eliminating anti-V2 interfering antibody might increase vaccine efficacy. And we set up to do this experiment in which we removed the V1 from the HIV envelope. However, as you know, proteins are very finicky. So if you actually delete some region of the protein, you might actually induce the degradation of the whole protein. So we collaborated with the protein engineering individual, Dr. Cardoso from the NYU, who helped us to really try to make a prediction by doing computer modeling on how we could eliminate the V1 while we maintain a good V2 structure. Because as I told you, you want to present the V2 to the immune system because the V2 is eliciting protective antibodies. So what Dr. Cardoso suggested or saw when he did the modeling is that, well, if you take off the V1, you can actually induce two different conformations of V2. One which is defined here as the alpha helix, and the other one is that is defined as the Greek key or beta barrel. So we didn't know which one would be protective. So we decided to actually generate a vaccine for both. And we deleted the V1 in a way that we could maintain an alpha helix or a beta barrel conformation of V2, as you can see here on the right. But we based that on the SIV because our model is based on the simian immunodeficiency virus. So here is the study that we did. And what you can see in here is on the top, you have a group of animals which were immunized with the wild type immunogen, which is known to have the V2 in both conformation, the green and the red. Then we also immunized them with the delta V1 that would favor the alpha helix conformation, the green, or that would favor the red conformation, which is the beta sheet. And the results were relatively clear because we got protection only with the, when we used the alpha helix. And what was important is that not only we got protection only in this group, but in this group, there was also a correlation with the immune response, the cytocidal V2, this is an ADCC, which stands for antibody-mediated cytotoxicity. And you can see in here that you have a very nice correlation with the time of acquisition, which really represents the risk of SIV acquisition and this immune response. Also, we did some other studies in which we took a monoclonal antibody that were able to block the alpha helix of the V2, cut off the FC fragment, and then ask the question, what happens if we compete this activity? Do we still see, can we compete it? And because if you can, then you can actually prove that some of it is really V2 specific. And that's what you see here on the right. And you can see that the difference between the presence or absence of the competing FAB is actually the one that still correlates, suggesting with the risk of acquisition, suggesting that it is the V2 specific ADCC that is actually correlating with the decreased risk of acquisition. Now, people in our field would be critical of this assay because it's done, is a kind of an assay in which we take, we take cells and we coat them with the GP120, and then we ask the blood cells of animals in the presence of the antibodies to kill the target cells. So it is a little bit of an artificial system. So we wanted to make sure that what we were seeing was really relevant also to the infected cells. So we took SIV infected cells and repeated the assay and asked the question, do we still see the correlation? And the answer is yes, as demonstrated here on the right. So the animals on the right, all these squares that you can see in there, which acquired the virus very late or never became infected, all had a higher level of this ADCC. So this makes us pretty confident that we do understand at least one of the mechanism of protection. So, as you know, science is about being able to reproduce the results. So we wanted to immediately, we repeated the experiment, and actually we have repeated that with another colleague. So we gave them the vaccine, and she was able to reproduce it together with us. And you can see in here that this was true not only in male, as I showed you before, but also in female. So a vaccine protocol, which is no more than 3 months, can now protect female. And here we had 65% vaccine efficacy. And it was also important to see whether the correlate was the same, and it was. So we look at the ADCC with that artificial, as I told you, more artificial assay in vitro, but it correlated. And when we looked also to other cells, which are monocytes, we also found that there was a correlation between the monocytes and the time of virus acquisition. I'm bringing this one up here. We published a paper on this showing that the ALVAC that is using here, the canarypox, is actually infecting monocytes and is able to induce hypoxia and inflammasome activation in monocytes. And the activation of these monocytes is really a key component of, I think, the ability of this vaccine to protect against infection. So, okay, so to summarize for you, and obviously this summarizes a variety of papers that we have published, but what I think what is happening in here is that we have a combination of vaccines in here that you can see in here, DNA, ALVAC, and alum, that are able to activate the inflammasome and produce IL-1 beta. IL beta is a very important cytokine that is able to induce emergency myelopoiesis. So the bone marrow starts to make make many more monocytes. These monocytes undergo epigenetic changes, and they cause either and they can cause anti-inflammatory monocytes, and also help the T cells to make V2 specific cytocidal antibodies. I believe that the reason why we are protecting in the mucosa while we vaccinate systemically our animals is because these anti-inflammatory monocytes then become macrophages. They can go in the tissue, and together with the antibodies, they might now be able to kill the infected cells. So an additional technology that we patented is about the activation of the RAS. What we found in large study is that the activation of RAS is actually really key, even if it is even key for protection. And in fact, we found that the animals that were able to already have a higher level of RAS activation were the animals that did better, and that this particular vaccine that we are using is able to further increase this. And this activation of RAS correlated nicely with some of the protective responses that we have seen in here. You can see in here is the top part are the different genes that were activated by the RAS pathway. And on the bottom, you see the little circle. For example, the stronger the color, the stronger is the correlation. And for example, the RAS is very important for the induction of antibodies to V2, but is also very important for the induction of anti-inflammatory monocytes. So here is just to give an example of what is known in the field is that these monocytes, which are cells that are, as I said, produced by the bone marrow, and every 22 hours, they actually die and they get replenished by the bone marrow in normal individuals. What happens is that when they see a pathogen stimulation, they undergo epigenetic changes, which means that their chromatin changes, and therefore all the transcription repertoire is now different in these cells. And also they live much longer. They don't die every 22 hours, but they actually live and they become macrophages and they go in the tissue. And the idea, and this has been shown in a variety of, for a variety of pathogens, is that when this pathogen is seen again, you can respond in two different ways. One is with so-called pro-inflammatory monocytes, which are making a lot of cytokines. One instead is, and this does happen in sepsis, for example, to limit the tissue damage, you can have a tolerant response, and you can make cytokines, which would then dampen the immune response to the virus to save the tissue. So we made a hypothesis, which was then confirmed by the data, that what we have in here is the second case, is the tolerance. And this makes actually sense because HIV, the reason why it has been so difficult to make a vaccine against HIV is because the virus is infecting the very same cells that are supposed to defend the host from its infection. So, and particularly CD4 cells. So if you are inducing too many CD4 cells, then what you are preparing a carpet of targets for the virus. And that's no good. This can actually affect you, even if you have a good vaccine, but if this vaccine is inducing also too many target cells for the virus, then you don't see the vaccine effect. And I think that our vaccine is doing exactly this, because the ALVAC is able to induce IL-10, it is known, the alum is able to induce IL-10. So the combination of these vaccines happens to be good in creating a tolerant immune response. And if a tolerant cellular immune response is in the presence of effector cells, which are able through antibodies to block the virus, is probably what we have in here. So we believe that by keeping in mind that we can activate the RAS, we could, so that, the activation of the RAS pathway could also be used to make even more effective vaccine. And okay, so the last, I just want to describe to you the combination of the vaccine. How did we arrive to put together the LVAC with the DNA? So the LVAC vector is a proprietary vector from Sanofi Aventis. We started this collaboration, as Dawn indicated, many, many years ago, 30 years ago, and we are still collaborating with them. And you can clone in this big pox virus vector a gene of interest, in this case, GP120, or also the remaining of some other protein of the virus. And when this was done, this was the protocol that was described before. It was done in Thailand, and there were 4 inoculation over 6 months with 4 inoculation of the recombinant LVAC. In this case, was HIV for humans. So we have done it with the HIV. And then 2 immunization with 2 proteins, 2 GP120. And with this protocol, there was an efficacy of around 31.2% in the 8,000 vaccinated individuals compared to the 8,000 placebo that they had in Thailand. And these results were reported in 2009. So by then, what we did is we recapitulated generating a very similar vaccine, but based on HIV, the results in the macaque. You can see here, this was true for following intrarectal challenge of animals. You can see that the vaccine has an effect. The vaccine efficacy was around 44%. It was also true in female macaque if we were challenging them vaginally, as you can see in here. But then we thought that we needed to improve it because having 30% vaccine efficacy or even 40 is not sufficient for FDA approval. So we tried a variety of things, which I don't have the time to take you through, and we had a lot of failure, actually. Many studies changed the adjuvant, we changed vector, we changed a variety of things. Anyway, to make the story short, I'm showing you only the good results. So the good results is that when we decreased the time of immunization and also the number of immunization, you can see here that we got a much better vaccine protection, probably because we don't elicit the CD4 cells that I tell you. We decrease probably the number of target cells. That's my hypothesis. We have some data that suggests that, but it's possible that that's the reason. So here I'm showing you this protocol in which we are still using 2 DNA instead of 2 LVAC, and then 2 LVAC and 2 GP120 protein on a period of 3 months. Now, just for the purpose of practicality, you know, producing DNA is much cheaper than producing LVAC. LVAC is able to grow only in chicken embryo fibroblasts. So you can imagine that to make the LVAC, you have to have these big roller bottles with chicken embryo fibroblasts to make enough virus. So it's much more expensive. So the DNA, the substitution of the DNA prime is actually really a cost-saving proposition, in addition of being also more efficacious, as you can see here. But then we further, and then when you look at the time of how long is this protection lasting, here I show you an example in which we took 11 animals that were protected from infection. We challenged them again at 7 months after the last immunization. So is here. So we had already challenged them and they resisted these 11 challenges, and they're done weekly. So weekly we exposed them to the virus. And then we did it again, almost 1.5 years later, and several of them were still protected. So the risk of acquisition, we are looking here at the risk of SIV acquisition. You can see here that it was still significantly maintained. So what are the potential development for all this technology? I think that combining the DNA LVAC with the modified V1 deleted envelope might be able to give us protection also in humans. And we want to test whether the immune response is similar in humans in this NCI phase 1 clinical trial that is in preparation now. And the GMP production of the proteins is, the delta V1 protein is ongoing, as well as of the DNA with the delta V1 in the envelope. And we also have ongoing the production of the ALBAC that the Sanofi has generated and will be used in the phase 1 clinical trial. So these are 3 independently effective technology, which when combined produce a more highly immunogenic HIV vaccine, having a deleted V1 region, modified GP120 immunogen. So at some level, we arrived to this conclusion a little bit by serendipity. The DNA and the ALBAC are all affecting the inflammasome and the monocytes. This in part is known, and also we have further confirmed it. So by combining them, I think we might be able to obtain a more effective HIV vaccine. So here I would like to now ask some of our collaborators in this from the Office of Technology Transfer to comment on these slides. If it is possible, let's see if I can get.
I'm here, Dr. Franchini.
Yes, perfect, okay.
All right. As mentioned, Dr. Franchini and her research team, they are seeking licensing as well as co-development research collaborations. The first technology, which is the V1 deleted envelope component, is undergoing patent prosecution. We will be entering the national stage soon for this in roughly April of next year. The second technology, which relates to the REST activating agent, has already issued. And also technology number 3, which relates to the use of DNA and the ALVAC vaccines, is also issued as a patent. You can find the PCT applications, the published form for each of the technologies, as well as the issued for technologies number 2 and number 3 can be found. It's publicly available. So my contact information, if you are interested in licensing or research collaborations, will be presented at the end of this presentation. And please relay any questions or interests you may have.
Thank you.
Thank you.
Okay, so we think that what we have here is by combining this technology, we will have an enhanced vaccine efficacy, and also by reducing the time and the number of immunization, and also the number of immunogen, because when we use the Delta V1, we don't use anymore 2 GP120, but only 1 GP120. So we have fewer vaccine component and reduced cost, also because it's a reduced time, and also a reduced time and perhaps increased ability to recruit and maintain volunteer when we do the trials. And another important point is that this vaccine have been already in many people. And the ALBAC has been not only 16,000 people, but also has been in infant. So the safety profile of this vector is excellent. And the DNA is also a very safe vaccine. And the alum has been used in probably most of the vaccine that we know of. So the current mark is clearly the potential vaccine users is anybody that could be exposed eventually to HIV. And the competitive advantage is that if the vaccine works, you don't have to worry about antiretroviral therapy, obviously. So as you can see on the right, the addressable market is the market that will generate people that are now free of HIV by using the vaccine. So in conclusion, I think that this vaccine, the Delta V1 V2 alpha helicase vaccine presented in the DNA poxvirus platform has the potential to protect against HIV infection and decrease the HIV prevalence worldwide. And all the components of this vaccine are safe and suitable for vaccine and adults. And I think it will be very helpful to have the results already of a phase 1 or perhaps even 2 clinical trial that is planned by the NCI for 2023 and 2024. So having this data already in humans, particularly if we can show that we can recapitulate the immune responses that we obtain in macaques, and I think is highly likely because the macaque immune system is really very similar to the human one. If we can do that in this model that has recapitulated also the results of other vaccine trials in humans, I think this would actually expedite the progress to the phase 3 trial, really try to understand whether a vaccine like this could be efficacious in humans and eradicate potentially HIV. So what we are looking for is a collaboration and licensing partner to assist with the NCI phase 2 and 3 clinical trial. As I said, the phase 1, we are already planning to do it, and it's going to be paid by the government. And this trial will involve the recombinant Delta V1 GP120 and the LVAC vector, and then take on the commercialization and bring this to the market. And I'll thank you for your attention. Here are the name of the people, Alkita Pollard that you have heard before, if you have any question, and as well as Wendy Patterson, as well as myself. Thank you for your attention.
Great, thank you so much, Dr. Genoveffa Franchini. Again, my name is Dawn Van Dam, and I'm the Executive Director of 1 Million Solutions in Health. So I would absolutely encourage you to put any questions that you have into the chat box now so that we can talk about them. Previously, the person who came on to talk about licensing was Rakita Pollard, who you see there at the top. So she is the technology transfer manager for this technology. So from a scientific perspective, if you have scientific questions, you could always contact Dr. Franchini, but if you're interested in licensing or working in a collaborative way on this technology, Rakita Picard is the person to contact. So let me jump in, Dr. Franchini, can you hear me okay?
Yes.
Okay, great, awesome. Let me jump in with the first question. How predictive for humans is the SIV-MACI model? Okay, so just to clarify further, I mentioned that at the beginning, but this model is, the SIV is genetically very, very similar to HIV in terms of its genetic regulation, and is causing disease in the same way, is actually affecting the gut, killing the CD4 cells. So from the pathogenesis point of view, the disease that the SIV causes in macaque is equivalent to the disease that HIV causes in humans. In fact, basically, even macaques develop what humans do in the presence of HIV, which is lymphoma, for example, and of course, immune deficiency. So having said that, there are still differences between SIV and HIV in terms of viruses. The sequence is different, but as I mentioned, there have been at this point 7 trials, HIV vaccine trials done in humans for a total of more than 40,000 people. And what the SIV model, this particular SIV model has done is really either to predict or recapitulate all of them. So to me, that's a reasonable, this gives me enough confidence to keep using this model to select some of the vaccine approaches that we have tested, and the adjuvant, for example. So we will continue because it'd be great if we could get even a better vaccine efficacy, for example, by changing adjuvant or even further tweaking the immunogen. One thing that I forgot to mention in my presentation is that we could also make the recombinant LVAC with the Delta V1 in it, because we have not done it yet, but that's something that is for the future. So I have confidence that this is a very reasonable model to predict what happens in humans. Great, thank you for that answer. Some of these questions came in, earlier, so you may have covered them, but I like to ask them nonetheless. And this one kind of builds on a statement you just made there. So, could the LVAC be substituted with the DNA?
Okay, so we don't know for sure. We are doing some experiment to try to do it, and the reason why we are doing that is because it would really decrease further the cost of the vaccine, and also simplify, all the licensing issue related to that, having an additional component. So we are doing an experiment now in which we are asking this question, and but because the DNA is less immunogenic overall than the ALVAC, and one of the reasons is because, you know, the ALVAC is a virus which can very quickly reach the cytoplasm of the cells, presents to the cytoplasm a lot of DNA, its own DNA, and that's why it's really very immunogenic. We can do the same with the DNA, but it's probably less effective. So we need to test it, and then to see whether what is possible. We have some preliminary data that suggests that if we just substitute the DNA as such, we might have vaccine protection, but it might be a little bit less durable. So we needed to test the different adjuvant, which is what we will be doing.
Great, thank you for that. Could you just go back to the slide with the patent timing on it for a moment?
Okay. One second. This one?
Okay, yes, thank you very much, great. The question is, taking note that technology number 3, the expiration of those patents is in 2021 or 2023. Is this component of the technology really necessary? Or do you see that the combination of the 3 is what we're really looking at here and making it therefore patent protected? Or how do you see that working?
Right. So this, as you said, is related to the prior question. So, you know, if we can avoid using the ALVAC, and we can substitute it with the modified DNA, that would resolve this issue. So we are doing that. We are doing the study to see whether it's possible.
Great, awesome. Okay, you can go back to the information slide in case someone wants to grab your contact information. Great, super. So a detailed question about monocytes. Monocytes are continually produced by bone marrow and live an average of 22 hours. Yeah. In your study, they are a correlate of risk. How do you explain the durability of the vaccine effect?
Okay, so there is a large body of literature that shows that following an insult, whether it is a pathogen or it is a vaccine or it is you know, a foreign substance in our body, we respond immediately with cells like monocytes, and the job of the monocytes is really to gobble up pathogens or a foreign substance, and also fix the tissue after that they've been received this insult of the invading pathogen. So it is true that the monocytes are made every 20, you know, daily basically, but it's also apparently true that some of them are able to go to the tissue and stay there for a very long time, if not for good. So the idea in here is that, and the idea that people have in the field is that vaccination through, for example, the production of a lot of cytokines and also chemokines that allow to these cells then to change and go to the tissue. And what is happening is you are changing the chromatin, you are creating a memory. And in the case of, for example, of the adaptive response, as we know, like antibodies or CD8 responses, this occurs through genetic recombination of either the T-cell receptor or the different immunoglobulin genes. But now people have discovered that also these innate responses, which are mediated by monocytes, are occurring not through genetic recombination, but through epigenetic modification, which are long-lasting. So the idea is that you have, let's say, a cytokine that is giving a signal to a monocyte, and the monocyte is now changing the way in which the chromatin is organized. So there is a loosening of the nucleosome, for example, I'll give you an example of, for example, the IL-10 genes, which is what we have studied. And then also what you can do, you can there is an assay which is called ATAC-seq. I didn't have the time to present all you this data, but we have them. That what this assay does is you take the cells, you purify, let's say, the monocytes, before and after vaccination. And then you take off the cytoplasm and you use only the nucleus, which is still live. You can take an enzyme, which is called transposase. It goes inside the nucleus and is able to target the region of the chromatin that are loose. When the region of the chromatin is loose, usually you have transcription. So you make the RNA. So there are methods to measure that. And we have seen that indeed that's what is happening in our vaccine. So the monocytes that these vaccines are inducing are not dying like most monocytes would do after they patrolled all the tissue if they don't find any pathogen, but they are actually responding to the immune response of the vaccine. They are one of the immune response to the vaccine. So they change epigenetically, and presumably, we have not proven formally yet, but presumably you have to assume that they stay in the tissue, because if they weren't, why would they correlate if then they are not there anymore? Most likely they are there, but they are now macrophages in the tissue. So I know that is not a complete answer, but is actually a working hypothesis. And in other systems, people have demonstrated that. We have not demonstrated yet, but we will do it in the next few years, I hope.
Great, awesome. And I'm sure you answered this question, but just in case there's anything else you want to say additionally, what do you think is the monocyte's function? Or do you feel you've covered that all?
So I think that it is known that monocytes, which are anti-inflammatory, are able to phagocyte, so to eat, basically, and to destroy pathogens. Or in cells, for example, also cells, they can incorporate cells and kill them. So the assay, the so-called, what I define as a little bit artificial assay that we used, which correlate with protection, which is called ADCC, the antibody-mediated cytotoxicity, is an assay actually that picks up not only the ability of natural killer cells, which are a different kind of cells, to kill the infected cells, but also the ability of the monocytes then to eat up the debris of the infected cells, of the killed cells. So I think that the function of these monocytes is to do that, to clean up and fix the tissue after the virus has invaded. I don't believe that we have what is defined sterilizing immunity. I think what we have here is that the virus can infect but in the mucosa, because we are giving the virus in the mucosa, not systemically. And these monocytes are there in the presence of antibodies, are able to, together with the NK, probably just to clear the infected cells.
Super. Two things. I have a number of more questions, but one is I'm just going to ask you to start your video again, because you're so very passionate about all this that I just love everybody to see your energy and things like that.
Oh, okay.
And the second thing is I'm going to be sending in the chat box a link to a survey that we would love everybody to fill out, just quickly giving some feedback on the science, technology, and this webinar. So that would be great if you could all help us out with that. So thank you, Dr. Franchini. Great to see you in person. So another question for you here is, what is the timeline for the phase 1 trial at the NCI?
So the product have been started, have been started already. And so the timeline that they have is 2023, probably. You know, of course, sometimes you cannot really predict when you try to develop biological material that needs to go to humans. You have to have a lot of rules and regulation and quality control and so forth. So, but the prediction would be, you know, 3 years from now, that would be, we should be able to start the human trial.
Okay, great. Thank you for that. Do you feel the vaccine regimen could be further simplified?
Yeah, I think it deserves some effort to do it. I mean, as I said, you know, if we could simplify it and get rid of the ALVAC, that would be useful because it would become much cheaper, more affordable, and simpler also to produce. But also, we don't know whether you need all those immunizations. So we started from those because early on, very early on, 30 years ago, when we started to do all these studies, it was so difficult to protect against any viral infection in macaques. And one of the reasons was because we were using too much virus to challenge. When science informed all of us that, HIV is infectious, but you need, usually the transmission doesn't occur with millions of viral particles that are infectious. So when we tuned down a little bit, tuned better the macaque model, we started to see protection. But at that point, we were already using a lot of immunization because we were frustrated by the fact that we couldn't protect. So this is a little historical perspective on why we have this very complicated and numerous immunization. In the case of HIV, if you look now at the coronavirus, 2 of them are enough. And for many viruses, 1 is enough, right? So I think we needed to go back and revisit that now that we have a vaccine that seems to be working. So we couldn't do it before, right? Now we can. Now we can say, okay, do I really need the second immunization of with the DNA, for example, or can I do without? Can I do with only 1? And so forth. So this is something that my lab will continue to investigate.
Great, thank you for that again. It's just a question around safety. Can you comment about the safety of the vaccine and why you think it is so safe?
Okay.
Like fundamentally why?
Yes, so there are evidence in humans that these are safe. But you're asking me why I think it's safe. I think it's safe because the heavy pox, the ALVAC is a canary pox. So it's obtained from canaries, has never been in humans naturally. So what happened is that the virus, which is a very large DNA virus, has never evolved like a vaccinia, for example, the vaccinia derivative, because vaccinia has been in humans, smallpox has been in humans. They have developed, the virus has selected, by being in humans, a bunch of genes, and some of them, in fact, even borrowed from eukaryotes, genes that were able to block the immune response. Instead, the ALVAC doesn't have that, or at least has it at a much lower extent. So what happened is that you infect the cells, but the virus can't really go ahead in its replication. So in fact, what we do is here, what Sanofi did when we generated the vectors was to use a very early promoter of the virus, because if you use a late promoter, you don't really express much of your heterologous gene in it. So in other words, the virus undergoes an abortive infection and therefore is safe.
Okay, thank you for that. One of the things that really struck me in our preparatory conversation around your science is sort of this different direction that you've taken versus maybe some of the other organizations that are looking at HIV vaccines. And the piece that was really, quite striking, I think, was that you weren't just looking at what was working, but you looked at what did not work and why. So could you maybe just explain a little bit about that thought process and how you got to where you are today from that?
Yes, actually we found, as the technology number 1 is an example of how you can learn more from something that doesn't work. That's something that does work. So, because, being in the government and having access, and I'm very fortunate having access to quite some resources and having the possibility of having a stable budget. So we were able to do these studies over the years using always the same model, the same viral challenge, which is logistically is complicated. But if you have the same operator, if you have the same facility, you can now do a larger number of studies with larger number of animals. And then you learn as much from the animals that are protected as from those that are not. So basically the so-called negative data are very informative if you are willing to dig deep. into why things didn't work.
And the difference in the scientific principle would be that you found by looking at the negative results?
Yeah, so the scientific principle. So to give an example, some time ago, before the result of the V144, people were making vaccines in which they took off the V2 region. They didn't know, right, that the V2 is important. Then the V2 was added on, but then, for example, sorry, let me go back. This is because this is a complicated question. Let me give you an example. So an example is they've done their V144, which is the trial in Thailand, with 16,000 people. And they saw that there was a sign that the vaccine might be possible. Then what did the field do? What they did is they went back and they organized another trial in South Africa, and using a different insert, obviously, because in South Africa you have a different clade of HIV. And in there, because they, for a variety of reasons, because, the collaboration that the companies establish among themselves and whatever, but what happened is that they changed the adjuvant. And by changing the adjuvant, they changed totally the immune response, and that vaccine didn't protect. So many people have said, okay, then this vaccine doesn't protect. But actually the animal data are saying the opposite. They're saying that you shouldn't give up after you have done experiments which were not really fully controlled. So again, we have the fortune at the NIH to be able to do more controlled experiments in animals. And these have been very informative, telling us what could be working, what are the mechanisms, and perhaps in which direction we should be going. So I hope that I'm correct in this, in terms of really thinking this.
That's awesome. Thank you so much. So I'm just going to do one last call for questions. I think we're at the top of the hour, and I think we've gotten through all the questions that have come in. So thank you very much to everyone who sent those questions in. That was terrific and really helped to explore a number of different areas that were important to cover today. So Dr. Franchini, maybe you could give us just a closing summary on why you think that this is a standout technology for HIV vaccination.
I think that it is because this is it goes against the grain of thinking that the more immunogenic vaccines can be better, and this has been really what has led the field of HIV vaccine. And our data are actually suggesting instead that you need to create more of an anti-inflammatory landscape, immunological landscape, to protect against HIV. And I think this is probably the novelty that we have in here, in addition to, to the construct that helped us and the vaccine that helped us to demonstrate this. So I think that we are making, I wouldn't say a leap of faith, we are just following the scientific results and try to see whether what we see in non-human primate is also true in humans. And because if it is, what we have at stake is really very important, because we could really prevent infection in particularly, in everybody, but particularly, and that's why we really are working a lot also with vaginal exposure, particularly vulnerable young female as well as children.
Great, awesome. Well, thank you so very much for your presentation today. Let's all remotely give Dr. Franchini and her full team a great big round of applause. And thank you so much for ending out 2020 with an awesome technology, a very huge opportunity that still needs to be solved in many, many areas of the world. And thank you for persevering and finding a way to make an awesome and very good vaccine. So again, here's the contact information for Dr. Franchini and for Rikita Pollard if you'd like to find out more information. And we look forward to much success for you, Dr. Franchini.
Thank you. Thank you very much. Bye-bye.
Our pleasure. Bye now, everyone. Have a great holiday season. Take care. Bye now.